Hello, learners. My name is Rick Myers. I'm at the Hudson Alpha Institute for Biotechnology, a nonprofit research and teaching institute in Huntsville, Alabama. I'm happy to be giving this lecture, sad for the occasion. I knew Casey Brown quite well. He rotated in my lab during his graduate work at Stanford. And then I was on his dissertation committee and kept in touch with him for quite a while. Anyway, I'm happy that we're able to honor him with this lecture series. So I'm going to talk about a little bit of history here, actually. I'm old, so I'm trying to kind of look back on how our field not just got started, but the Human Genome Project, the beginnings of that, and what we did in order to get DNA sequence, and then what we've done with it since. And I know people are interested in lots of different aspects of this, and that's what I'm going to cover. Let's see. So I've been interested in, for the last really 40 years or more, of trying to find genes that are involved in important phenotypes. I happen to work a lot on human brain disorders, neuropsychiatric and neurodegenerative disorders, as well as sort of the basic biology that you learn from finding and then studying these genes. And then really, it's one thing to find a gene, it's another thing to then figure out what they do. And when we talk about a disease gene, it's a little bit of a misnomer because genes, we all have the same genes. It's just that sometimes there are mutations in them that actually lead to phenotypes, mostly disease, but some are a lot of those are diseases, but a lot of them are other traits as well that are interesting. And so it's obvious that DNA sequencing matters a lot in this. It's not the only tool we or others use, but being able to determine the nucleotide sequence of DNA and actually RNA, of course, are really important for this and much of biology. So I'm going to spend most of the time talking about sort of the DNA sequencing technologies, but then some applications. So obviously, DNA is the informational molecule. And what we want to know is the sequence, of course, is the order of the bases A's, G's, C's, and T's along the length of the helix. We'd like to know that for everything, for all of the genes and all of the genome. And just imagine most of you know this, but let me give an example of why this is important. And this is an old example, one that we've known the answer to for a long time. And that's sickle cell disease. Used to be called sickle cell anemia. It's much more, has many, many phenotypes in addition to anemia. And interestingly, this is due to a single base pair change at one position out of all of the 3 billion base pairs in the human genome. So if you look at, and it codes for beta globin, one of the two hemoglobin proteins. And if you look at this on amino acid 6, there's a GAG codon for glutamate, and that's what most people have. But if you have a mutation where the A is changed to a T, you get a valine coded there. And that one change in the protein, you still make the protein, but you make an aberrant protein that leads to serious phenotypes. And it's called sickle because the normal flattened, round red blood cells, which go through the capillaries, almost single file, to release oxygen and take up CO2. They sickle, and they go through cycles of turning into a sickle shape, and that actually just physically blocks the capillaries, and that's what causes the phenotype. So really important to be able to find the sequences of genes as well as the sequences of the mutations that can cause a phenotype. So I'm not going to go through all the early history. Don't worry. There's some really interesting history, even pre-1965. But the first time a nucleic acid was sequenced was a fellow named Robert Hawley and his colleagues, who essentially sequenced a small tRNA molecule, it's 77 nucleotides long, and won the Nobel Prize for this, by the way. But he did this by sort of a chemical method. You cut the RNA, you purify the RNA, you cut it into small fragments, and these are overlapping fragments. You do use different kinds of enzymes to cut them. And one of the enzymes might cut after an A, for instance, another one after a G. And by doing that, he was able to take those small fragments, do some chemical work to figure out what they were composed of, and then was able to figure out the order. This was a 3-year-long project, and it was a big deal because it had not been done at all. And then 10 or 12 years later, Fred Sanger and Alan Maxim and Wally Gilbert developed 2 independent methods, independent of each other, but 2 very different methods. And one of them is called Sanger sequencing. Fred Sanger won his 2nd Nobel Prize for this. He had done protein sequencing before that. And it's a remarkable method that still was used throughout the Genome Project, as you'll see in a minute. But it was very crude in 1977, and I'll show you what the experiments looked like. Maxim and Gilbert did something that had some similar principles of measuring the lengths of fragments on a polyacrylamide gel, but it was a chemical breakage method, also Nobel Prize given for this one. So Sanger sequencing is what we ended up using the most. The Maxim Gilbert, I did a lot of that in graduate school. We use it for other reasons as well, but it's dangerous chemicals and harder to get to work. But the Sanger sequencing, basically you take DNA molecules that you want to sequence, and these were for the most of the time have been DNA clones, plasmid clones that you clone into E. coli and then have a large number of DNA of the same DNA molecule. You basically you use something. You use a DNA primer, and you use it to extend the DNA that you want to sequence with 4 different reactions. One of them will stop at an A, one will stop at a G, one will stop at a C, et cetera, and a T. And you set those up so that it doesn't stop at every T, it's or C or A or G, but you do this and grow the chain. And then these are labeled in some way or another. We used to label with radioactivity and then later with dyes. And then you measure the lengths of those fragments that are left over from the molecule on a polyacrylamide gel. And basically it's measuring it based on size. So here's a sequencing autoradiogram. This was P32 or S35, I don't remember, but what was radioactive. And you basically, like you do in this little cartoon, you read in the G lane, it goes G, et cetera. And we did that manually, and it was a real pain. And this gel that I ran here, I did maybe 4 of those in one day, and that was a massive amount of work to be able to do it for getting just a few hundred base pairs, and not very accurate, I should say. It was okay, but the electrophoresis was sometimes wonky, and it would make it not perfect. So you would sequence it multiple times sometimes to try to get it accurate. So that's the way it stood in the mid-1980s. And the idea came about of, why don't we figure out how to sequence not just the occasional gene here that we're interested in humans or any other organism for that matter, but try to sequence the entire human genome. So let me I happened to be at the right place at the wrong time. It was almost, I mean, right place at the right time, excuse me, that almost by accident. There was a meeting held by the Department of Energy, the U.S. Department of Energy in Alta, Utah. And they wanted to know, could you figure out the germline mutation rate in people? Meaning, how many mutations does a child have that neither parent has? Something happened during sperm or egg development that would cause a germline mutation. And they held this in Alta, Utah. My boss, who was postdoc boss, was Tom Aniatis, was invited, but Tom sent me instead. And it was a very small meeting, only 19 people. And basically, all these scientists who were doing this, they were not really genome scientists. We didn't even talk about genomics very much back then. They were doing various cell type assays. And somebody in the audience said, after a day or so in the meeting, the mutation rate is probably so low that we'd have to sequence the entire human genome to figure it out. And everybody laughed. This is 1984, because what an absurd idea. But the DOE folks they were really smart. They went back to their offices and said, hey, wait a minute, why don't we do this? So they came up with the idea of sequencing the entire human genome, uh, albeit knowing full well that it would be a really, really hard project to do. So the project was born, and at the beginning of the project, it started in 1990. I'll show a little bit of that in a second. But the beginning of that, we had 3 meetings, early, really late 80s, early 1990s, where we talked about all the people who might contribute to this project. Not all, but there were people from a lot of different labs in Europe and in Japan in particular, as well as the US. And we came up with the principle that if we're going to do this, it's going to cost a lot of money, and we can't do business as usual. You need to generate data and then release it to the public rapidly with no strings attached. And sure enough, we did it. We came up with the Bermuda principles. These were later, many years later, even altered a little bit more to make it even freer, called the Fort Lauderdale principles. But the idea was that this was a whiteboard that one of the folks at the meeting had written on. It turned out to be a really important thing. And I'm really pleased that this not only took, but it essentially is still essentially the approach that we take when we're sequencing large amounts of genomic DNA, humans or otherwise, that is rapidly released. So we began in October 1990. There were literally 2,000 or 3,000 scientists overall over all the years. It took us 13 years to do this. And the idea was to determine the order of these base pairs throughout our chromosomes, essentially from one person. It wasn't really one person, it was a composite, but essentially one human genome. Of course, you call this order the sequence when we sequence DNA or RNA or proteins for that matter. You're figuring out the order of the building blocks along the length of the, in this case, the chromosomes. And that's a lot. I mean, remember that slide I showed with where I sequenced a couple hundred base pairs over really more than one day. And manually analyze it for quite a long time. It would have taken forever to do the genome project with that technology. So we knew that the technologies had to get better, and they did. One thing I will say is that even from the beginning, we sequenced other organisms. Some of these organisms with smaller genomes, like microbes and C. elegans, and and yeast, for instance, had much smaller genomes. They were good to help figure out the technologies and try to improve them. And I will say that those technologies continue to improve throughout the entire project and beyond, and you'll hear a little bit about that. So we sequenced a whole bunch of other organisms along the way. The key to the human one was that we wanted to get a complete sequence of the whole genome. That happened with all of these organisms eventually, but some of that was getting drafts of those genomes. So the technology was improved enough by 1990 that we were able to do better than I was on that 1985, whatever, 82 gel that I showed, autoradiogram. So basically what we did, and I hope you can see my cursor here, was we took genomic DNA from one person, but it's really a composite person, sheared it into fairly large pieces, 200 or 300 kilobase pairs, some even longer, some a little shorter. So these were then cloned into a vector called, and you call these clones back libraries. We still use back libraries occasionally. They were hard to make, but once this was made, we then took all of these clones and actually organized them into a map along the lengths of each chromosome, as much as we could. I'm going to show a little bit about how that was done. Once the back clones were made, you then took each one of those individually, shared it into much smaller pieces, a few thousand base pairs long. Usually these were about 3 kb, 3,000 base pairs. Clone them into plasmids, and then made libraries from those. And this was millions of these clones. Then we sequenced each of those individual clones with Sanger sequencing. The gels were not radioactive. By 1990, there was this technology came out of Caltech that was 4-color fluorescence. So they were fluorescent dyes. It made it a whole lot easier to read and collect the data more automatically rather than reading them manually. And actually, this is from Marco Mara, who's in Vancouver. We map the back clones by sequencing the ends of them and then also doing restriction mapping on here. You cut it with restriction enzymes and then map them. So we could try to make what we call these large clone contigs where they overlap some, but not too much. Sequencing was still so expensive, it was going to be so laborious. We wanted to minimize the total amount that we had to do. About 7, 8 years into the project, one of the members of the genome, the public effort, this was the free release of data, et cetera, started a company or came into a company called Celera, a new company, and they wanted to do it themselves, and they did. And they did a good job on it, but it was a different technology that actually many people were concerned might not work, but it turns out it works really well. You take genomic DNA, all the chromosomes, it's just a mixture. You fragment it, and these are now fragmented into small-ish fragments. And you clone those, and then you use Sanger sequencing and do the same thing where you get the sequences of the clones and then overlap them in the computer. Now that sounds almost identical, except instead of making a mapped version of all of these clones or the big back clones that we made, they just shotgun sequenced the whole thing. And there was some concern by the public effort that it would be really hard to do the assembly when there was so much assemble in the computers when there were so many fragments, and it was hard. But Celera, the team at Celera, led by a computational fellow named Eugene Myers or Gene Myers, was able to figure out how to do this, and it was quite remarkable. They put it together, and so what happened is we did that. And the two groups separately, but simultaneously, we made arrangements to do this, sequenced what we call drafts of the human genome. And I'm going to tell you what a draft means. It was really valuable, but it wasn't complete by any stretch. We still had lots of gaps and lots of problems. So the public effort of the Human Genome Project was called the International Human Genome Sequencing Consortium. The public effort continued on for 2 more years and did what we called finishing, basically trying to fill in as many of the gaps and getting the chromosomes where they were what we called a complete sequence. It turns out it wasn't complete, but it was close to it. And you'll see what I mean in a minute. We made this coincide with the 50th anniversary of the publication of the first structure of the DNA double helix, April 25th, 2003. A lot of celebration. It was exciting to have this done. Again, this took several thousand scientists, 13 years, and a lot of money. Much of the money was spent in making the clones and the maps and the setting up the infrastructure and doing the computing and all of that. But a lot of it was because we really didn't know how to do this at the beginning, and it took a lot of technology development. So let me now compare that to what we do now. And I'm jumping way ahead because after 2003, the technologies, we knew this was valuable to have one genome. So let's get as many as we can. And so it had to improve even more. And so a completely new approach came about called next-gen sequencing or next-generation sequencing, NGS. You'll see that a lot if you have it. And it's a little bit of a misnomer also because we're probably on the 13th version of next generation sequencing. The machines get better, the approaches get better, but the general principle is you take genomic DNA, it says cut here, we usually shear it randomly, and you do something to the ends of those DNA fragments so they have little places for oligonucleotides to land. And the huge difference here, instead of cloning these or even shotgun sequencing these directly as clones, where you make bacterial clones and you have microtiter plates with wells that you get millions and millions of such clones. Instead of doing that, you take that, what we call this a library now, once you've added the linkers on the end. You take those fragments and you just throw them onto a glass surface. It's not a simple glass surface. And they're the size of a microscope slide. They've gotten bigger now, and they had lanes, so you would throw them on. And remember, DNA is really tiny. You don't see it. It's very small. You land them on this glass surface, and you cause them to stick, and then you cause them to loop over or bend to the next station on the slide, and then you amplify this. So you get these clusters. Each one of those represents one of those DNA molecules. And then you do a type of sequencing similar to what, not Sanger, but something like that, where you're incorporating dyes, and then you get these images. This image is actually quite a tiny part of one of these glass slides. And at the beginning of next-gen sequencing, we would sequence a few million molecules off of one of these flow cells. And you'll see in a second, we do a whole lot more. Now we can, when you land them on these slides, you get literally billions of them landing on a slide, and you can run 2 slides on the sequencing machine. So now you're sequencing a huge amount of DNA on a tiny area where you didn't make clones, you didn't have to address the individual fragments that you were sequencing or grow them up and put them somewhere else. You basically do it by randomly landing things on a slide. So now we've sequenced billions of them, and it takes about a day to run these, the fragments on the sequencing machine. We call this short read DNA sequencing because the fragments that we sequence are 3, maybe 400 base pairs, and you're sequencing those over and over and over, redundantly, randomly sheared, redundantly. We call this 30x or so. We want to sequence the genome about 30-fold over so that we actually get as much overlap as possible, and then use computation to put it together. A lot of effort went into developing algorithms and methods, and they are constantly improving to actually do that assembly. And then the hard work comes in. Okay, so now I've got to assemble. What does it actually mean? And I'll talk about a little bit of that in a minute. So what do we have now? I actually don't know how many human genomes that are published, are completely sequenced, but there's several million. There may be as many as 10 million. We don't. Most of those are made available through these large databases and these big projects. Genome UK is one. The All of Us project in the US is one. But many labs do this for many reasons. And I'm talking about just human genomes there. And that usually has to do with understanding disease, understanding basic biology, understanding our evolution, understanding, population issues and things like that. We now have thousands of species of plants, animals, fungi, and protozoa. And actually, a lot of the plant genome sequencing is done here at my institute, Hudson Alpha Institute, by some of our faculty who were part of who came to Hudson Alpha from Stanford, who were part of the Human Genome Project that we participated in. And plants are hard, it turns out. There are a lot of repetitive sequences, but really valuable for things like sustainable agriculture, climate, making products from plants, et cetera. But so are animals and other organisms as well. And we don't even really know how many species of bacteria and archaea there are, but hundreds and hundreds of thousands of these are published and are sequenced data release, and people use this for all sorts of reasons. And then there's another approach that started about 10 or 15 years ago called metagenomics, where you take a sample from, let's say, soil or just off of our skin or something, and you sequence everything that's there. You're not purifying different organisms. It's been incredibly valuable to have these metagenomes or these sequences from various places, because you learn a whole lot and can use that for mitigating problems in the environment, et cetera. So I mentioned something about the draft sequence and 2 sequences, because we had both the Celera sequence and the public effort sequences. So let me compare that to 2 years later when we did a finished sequence. And I put finished in quotation marks, and you'll see why in a minute. So basically, when we did the draft, both of the public efforts ones looks like the top of this line. It was 2.6 billion base pairs. We thought we had about 92% of the euchromatin, not the repetitive telomeres and centromeres, et cetera. And there are a whole bunch of gaps. We all knew that, but it was still valuable to have that. When we did the finished build in 2003. It was July, well, really April 2003. It was, we had most of the euchromatin, we thought. It turns out we didn't. And we're still missing, a fair amount because of all the repetitive sequences and, a much smaller number of gaps. This project then continued, it's called the Genome Reference Consortium. This one sequence, we call it the reference sequence. It continued to improve by just taking that particular those particular genomic resources for that one composite person and improve it. So it got better and better over the years. But then, and you may have read about this, it got a lot of fanfare, as it should. It was an important project. About 100 people, many of whom had been part of the initial genome public effort for the Genome Project, did what they called the first really complete gapless sequence of the human genome. They called that the telomere to telomere consortium. They published this in Science in 2022, and there are a whole bunch of papers associated with it. And this was really hard. They basically resequenced the entire human genome from multiple people, it turns out. And they use something called long read sequencing, which means, and all that is similar to the other kind of sequencing, except the machines are different. But you're now sequencing end-to-end fragments of DNA that are as long as many tens of kb, maybe ranging from 8 or 10 kb to several tens of kb. And now you're sequencing continuously across repeats that were hard to assemble from short read sequencing. So they did a combination of these and a whole bunch of other experiments that then filled up in all the gaps. Now they needed to put complete in quotation marks too. There's some parts of the Y chromosome that have just been intractable or really hard that will probably eventually be done, but they were not completely done on this paper, although they covered a lot of it. So complete coverage of telomeres and centromeres are really repetitive, and they're long repetitive segments, but they're repeats throughout the whole human genome. We got through the small allo repeats and things like that in the initial Human Genome Project because with short read sequencing, but it's really hard to go through ones that are much longer, and some of these are tens of kb long. So they put all of that together, and proving that this was important, they found many new genes, a couple million more sequence variants, places in the genome where it varied from person to person, lots of regulatory elements, many, many things. So this is a great resource, also freely available, just like the initial human genome was. So let me talk a little bit about DNA sequence variation. You will have learned about or maybe know a lot about mutations or even SNPs or polymorphisms, but we're not all the same. In our DNA sequence. No organism is completely the same. Even identical twins have identical twin sisters. Their DNA is identical, except for the new mutations that occurred during the development of the germ cells that formed them. So any two people's genomes are almost like 1 in 1,000 places in the genome you vary. If you count the large structural variation places, which were much harder to measure, there's more difference, but it's still, look at this, it's only 0.4% difference, 99.6% identical. Interesting thing about this is true everywhere on the planet for all humans. You find the same variation in every population. We don't use the word race anymore because it's not really races. Race is a continuum. We call it ethnicity or continental origin. And you look at people from Asia, from Africa, from Europe, we all have the same variants, or all have many, many different variants in sequence, about every 1,000 base pairs or a little bit more maybe. But the frequency of those variants varies from population to population, and that's where you get some of the phenotypic variation. If you think about this, is also true of you. Anybody listening to this? We all have two copies, a copy from mom and a copy from dad, and those are not identical by any stretch. And so we each have 3 to 6 million differences between our two copies. There's a lot of differences. It's still a small amount compared to the 6 billion base pairs we're each carrying, one 3 billion from mom, 1 3 billion from dad. Interestingly, I don't really like to use the word neutral. In fact, there are all sorts of discussions about what does that actually mean. But most of these variants, some of them are in the coding regions of genes, some of them are in regulatory elements, some of them are in between, and there's a lot of DNA sequence in between genes and their regulatory elements. We don't really know what all of them do, but most of them don't have any discernible effect on phenotype or fitness, or we just haven't figured out how to measure the phenotype differences. All right, now I'm going to talk about using this information. I kind of think there are really more than two major goals of human genetics, but I like to think about finding the DNA sequence variants that affect phenotypes. One way to say that is, let's find the mutations that cause disease. And really what it means is find the genes that have mutations in them, because we don't know that for a lot of for many things. We certainly didn't know it at the beginning of the Human Genome Project in 1990. We probably knew mutations in about 70 genes that cause phenotypes, like sickle cell disease was one of those, and a few other ones that are particularly ones having to do with metabolic problems that were easily measured. So one of the reasons for the Genome Project is to find those genes, and really, it's really one of the reasons that biologists do this in human genetics and take genetics of any other organism, the idea of understanding these variants. And then another big part, and this is where much of other technologies, other disciplines really in biology come, is figuring out why do those sequences cause phenotypes. I work on most people work on disease phenotypes, but there are some really interesting other phenotypes like skin color, red hairs due to mutation in a single gene, for instance. Understanding things like that are interesting, but they also, you also sometimes have mutations that actually make you resistant to disease. We want to know all of those things. So I'm going to give, this is a pretty extensive example. It's a disease called progressive myoclonus epilepsy of the Verreck-Lundborg type, or also abbreviated as EPM1. It was found at a higher frequency in the Baltic states and Finland. So we collaborated with Finnish collaborators when we actually found this gene. This is old. We found this in 1996 when the Genome Project had was just starting to get sequences. Pretty bad disease. Kids start to show symptoms when they're 6 or so, and maybe a little later than that. There's mild but progressive cognitive decline, but the big symptoms are severe grand mal seizures, myoclonus, this jerking motion, and then ataxia. They're wheelchair bound because they can't keep their balance or walk. It really requires lifelong care. It used to be fatal. Now, because of understanding it and particular care and treatments, they live a much higher quality life. It's about 1 in 20,000 worldwide, which doesn't sound like a lot, but that adds up to a lot when you go around the world. And it's inherited, so we knew that it ran in families. And it was recessive, just like Mendel. We knew it's a Mendelian disorder. Just like Mendel, we knew that you had to have mutations in both copies of the gene. So back in really in the early 80s, all the way through the 90s, we would understand where genes that were simple genetics, meaning one gene is causative or is a Mendelian disorder, and there are thousands of those. We could figure out where those genes are located in our chromosomes by doing genetic linkage mapping, and you may have learned about this, but it's basically looking at the different polymorphic DNA markers, and we really only did this with a few hundred markers spread throughout the genome, that you figure out which polymorphic markers track with the disease in families. And really it required multiple families, usually a dozen families or so. And the Finnish group we collaborated with, Anna-Leena Lehtojoki led that group, found that it was on chromosome 21, on the long arm of chromosome 21. But that was still located to a region of several million base pairs. So you didn't just, and back then, figuring out where the genes lie in that was non-trivial. That's partly why we sequenced the human genome. But we found the gene by localizing on finding DNA clones that cover this couple million base pairs or a few million base pairs on chromosome 21. And then we we figured out which ones of those fragments, DNA clones, had expressed messenger RNAs. In other words, we looked to see where RNA is coming from them in human cells, and then we compared that to people who did not have the disease and people with the disease. We sequenced those to figure out, could we find mutations? So we found it. Len Pinocchio in my lab led this group. He was a graduate student in my lab, did this project with these collaborators. And we were lucky, it turned out to be a small gene, a couple thousand, a few thousand base pairs, and made it easier to actually analyze it and sequence it. And it has 3 exons. And basically by sequencing and comparing controls and patients, we did this on maybe a couple hundred controls, meaning people without the disease or without the disease in their family, and then patients, and actually the patient's parents as well. And we found initially 2 mutations and then ultimately a whole bunch of different mutations in these. And you can look at mutations, imagine you know this, you may have heard it, you can look at base pair changes in some places, and you know if you form a stop codon, that's probably going to be bad for the gene. There's several other types as well. They're right at the edge of splice sites. If you have a change, you can change the ability to make a spliced RNA. And you have missense mutations that you might even predict, going from a charge to a neutral amino acid might be bad for the protein. And interestingly, the most common mutation that we found was actually in the promoter, just right upstream from the start site of transcription. There's a repeat that would show up in patients as many, many copies as opposed to 2 or 3. That was predicted and proven that it's really bad news for a gene if you mess up the promoter. So that's what we learned in 1996 and published the paper. And the question then is, all that evidence that I just said was pretty strong. This is almost surely the gene. Sequencing a couple hundred controls for a lot of diseases is not necessarily going to be good enough. We do this much differently now. But what we decided to do for a lot of reasons is to follow up with functional studies. So as I showed in the last slide, they were loss of function mutations in both copies of the genes in patients, but not controls. When we looked at messenger RNA levels, and it turns out we could do this from blood RNA, just taking a blood sample from them, because the gene's expressed almost everywhere. The cystatin B, I didn't mention, this is the protein that is encoded by the gene. And the messenger RNA levels are at 50% level in the parents, because they have one good copy and one bad copy. All of these mutations cause the RNA to not be made. They were stop codons, you get nonsense-mediated decay, you get that in all but one of the mutations, I should say, certainly the promoter mutation. And I don't know if you can see the arrow here, but if you look at lane 1 and lane 9, this is a northern blot measuring the amounts of messenger RNA. You can see that there's slightly more in each of these 2 parents, and in each of these 2 parents, there's slightly less than the control, somebody who has no mutation. But the real smoking gun was the fact that the patients were completely missing the RNA, and that was true for other patients as well. That experiment alone was essentially evidence that how would you have that happen and all those other things happen without, and also being located in the right area of chromosome 21. So it was really important to nail this. But then we did something that many people do. We then made a mouse model. Essentially, mice have the same gene. They have the genes, many of the genes in the same order and very similar sequence. So we went in and knocked out Len Pinocchio, again, led this study, and knocked this out where we had mice missing both copies of the gene. And sure enough, they get seizures. And in fact, the seizures, we worked with Jeff Nobles and others as well to understand the types of seizures. The seizures are what you would expect, and they get myoclonic seizures and et cetera. They have ataxia. I won't go through this, but basically it's something called a rotor-rod experiments. How can they keep their balance on a rotating or a non-rotating rod? And sure enough, there was significant difference in the mutant mice as opposed to the wild type mice, and especially as they aged. And importantly, we knew this happened in patients, by the way, from a few postmortem studies that had been done decades earlier, that Purkinje cells, this type of neuron in the cerebellum, die. Turns out cells in the cortex die as well. But here we looked in the mice. Here you see these, I have the word granule cell, they're Purkinje cells. Sorry, that's a mistake. But Purkinje cells here, and they're gone in the mutant cells, are sorry, greatly diminished in the mutant cells. That's where ataxia comes from, because basically your cerebellum is needed for balance. So what? We did this. Does it do anything? Does it help anybody? And you would, of course, love this to lead to an understanding of the disease, to lead to new drug treatments or other kinds of treatments. You can certainly use them as diagnostic tools. You can try to do early detection so that you can start treatments earlier. There are all sorts of reasons for wanting to do this. Sure enough, that can happen here. The problem is there are lots of different forms of epilepsies where you get seizures. EPM1 is one of about a dozen different progressive epilepsies that children get. And they look very similar from one cause to another. In other words, the phenotype looks very similar. We now know some of the causes of some of the other ones, but when we found this, it was the first gene that was found in progressive epilepsies, or maybe any epilepsy. And so the neurologists have trouble distinguishing them. It's not their fault, it's just they look very similar. So as soon as, and then another fact that's really important. The physicians knew that a whole class of drugs, but especially the main one that was used that is given for epileptic seizures still is, it's called phenytoin or Dilantin. And it turns out it's extremely harmful to kids with this particular form of progressive epilepsy. In fact, it can be fatal as well. We don't really understand why that's the case. People are studying it, and they may have some better understanding now. But certainly, when we did this, it turns out the kids do respond reasonably well to another drug, and now multiple drugs. One of them is called valproic acid or valproate. So think about this. You don't know whether a family comes in and their child has a progressive form of epilepsy. You don't know what the cause is. And so if you try to use the common drug, then you're going to harm that child. And this did happen in the 1960s. And luckily, people started slowing down on it. But as soon as this gene was found, it was made public immediately. The physicians could use this as a diagnostic to say, is this EPM1 or is it one of the others? And if it's EPM1, you don't give them phenytoin. And sure enough, as soon as we did that, we were at Stanford when we did this. Stanford licensed this to a company called Athena Diagnostics, and they and subsequently others developed a test for this. It's actually fairly simple to test a single gene or even a panel of a bunch of genes, and they did panels here, and they would use this test. And I don't know how many times it was used, but when you think worldwide, 1 in 20,000, and in the US alone, there are 4 million children born every year. So there's always going to be new cases. So it's something that I'm very proud of, because here we are doing kind of basic research, but somewhat applied in trying to figure out the cause of a disease and study it, in the laboratory, that it then immediately has an application. This is one of many thousands of similar stories. There are lots and lots of examples of where something like this, where the treatment is different, or even you develop some treatment that was never even existed. This is particularly hard in brain-related diseases, and something that we work on with neurodegenerative and neuropsychiatric disorders. So finding disease genes today is very different. Actually, this is an underestimate. I found it's really hard to say how many Mendelian genes, how many single gene disorder genes have been found. It's well more than 5,000, because that's grown more and more. And some of these can be quite rare. So new ones are being found all the time. And again, public data, so people then use this to do things. But now we don't do it the way that I just described. We just sequence their genomes. And I say just because you can now do this for a few $100s really rapidly. Most of the effort going into handling the DNA and handling the data, interpreting it and disseminating it. But you sequence them and then now sequence from compare this to the millions of genomes that are available out there in the world, especially those data sets where we deeply know the phenotype, whether they have something or not, et cetera. And that turns out to be extremely powerful. And you may have heard, and if you haven't, you should watch Greg Cooper. Dr. Greg Cooper is one of my colleagues here, one of our faculty colleagues. He sequences thousands of mostly children, but a lot of adults as well, with undiagnosed diseases. We don't know the cause. Sequence these, and maybe shockingly, you figure out the answer in 30 to 45% of the cases. You figure out what the gene is, and then now, even if it's a really, really rare disease. So this is something that's being done in a lot of places around the world. People are sharing the data and making that much more rapidly finding what is the cause. If you have a really rare disease and you find a couple of people with mutations in a gene, you're probably right. Maybe certainly if you find a half a dozen, you're right. But then you share that information, and people around the world who've done similar kinds of things then share that, oh, we have mutations in that gene as well, and here's the phenotypes. That's been incredibly powerful for rare diseases, but then for common disease as well. So I appreciate you listening. I hope this was helpful. There's a lot more to go beyond just sequencing and then even finding the genes. I happen to study gene expression. I have been doing that since graduate school. We use genomic tools to actually understand not just the coding regions of genes, but the cis-acting regulatory sequences that actually control whether a gene's expressed and how levels of expression and which tissue is going to be expressed in. And that's a whole nother subject you'll probably hear about in some of the lectures in this series. Thank you.